We'll continue discussing about the Y chromosome and male development. Okay, we will discuss in detail how Y chromosome is responsible for male development. A current analysis of Y chromosomes has revealed presence of numerous genes. Okay, there are number of genes and regions with potential genetic function are present on Y chromosome. Okay, this is as per the current analysis, that is Y chromosomes have number of genes and regions with potential genetic function. Okay, same genes with or without homologous counterparts are seen on the X chromosome also. Okay, that is the X chromosome which is present along Y chromosome. It has same genes as that of Y chromosome with or without homologous counterparts. Okay, that is what are all present on the Y chromosomes are present or without also. Okay, the same genes with or without homologous counterparts are seen on the X chromosome. So this is the current analysis what it is revealing. Okay, and another information present at both the ends of the Y chromosome are the pseudo autosomal regions. Okay, which is in short it is called PARS, pseudo autosomal regions. Okay, so present at both the ends of the Y chromosome are these regions which are called PARS, that are similar with regions on the X chromosome also. Okay, the same regions PARS are seen on the X chromosome also. It refers to both the ends, okay, of the Y chromosome and X chromosome. Why? How come Y chromosome has got the regions? It is because of that is during meiosis recombination occurs. Okay, during crossing over which takes place at synapse. Okay, so due to that, Y chromosomes also have this PARS. Okay, pseudo autosomal regions. These are the similar regions as that of what are present on the Y chromosome. Okay, why this has happened? It is due to synapse and recombine during meiosis. The remaining 95% of the Y chromosome. Okay, apart from this PARS, the remaining 95% of the Y chromosome, it is referred as non-combining region of the Y, that is NRY. Okay, non-combining region of the Y, because it remains without any change and it is very specific only to Y chromosome. Okay, this NRY, it is not seen in X chromosome. Okay, so the remaining 95% of the Y chromosome, it is referred as NRY or non-combining region of the Y. Okay, we'll discuss now about the next one. So this NRY, okay, this non-combining region of the Y, it is divided equally into functional genes and non-functional genes. Okay, NRY, okay, that is divided equally into functional genes and non-functional genes. Functional genes referred as euchromatic and non-functional genes referred as heterochromatic. Within the euchromatic regions is a gene called sex determining region Y. Okay, within the euchromatin region. Okay, within the euchromatin region, there is a gene present called sex determining region Y, SRY. In humans, absence of Y chromosome inevitably, okay, that is compulsorily, this absence of Y chromosome in humans, it leads to female development. And this SRY gene is absent in X. Okay, sex determining region Y, that is SRY, it is absent in X chromosome. That is why the child is born as female. Okay, the gene product of SRY is the testis. Okay, so ultimately, that is what is produced from SRY that is the gene product of SRY. It is a factor which is called a testis determining factor, that is TDF, which is present in the adult male testis. Okay, and we shall discuss now about genic balance in Drosophila. It's a fruit fly. Okay, Drosophila is a fruit fly. And then genic balance mechanisms of sex determination in Drosophila was first studied by C.B. Bridges. Genic balance mechanisms of sex determination in Drosophila was first studied by C.B. Bridges. In Drosophila, the presence of Y chromosome is essential for the fertility of the male sex. Okay, the presence of Y chromosome is essential for the fertility of the male sex. But at the same time, it does not determine the male sex. Y chromosome is very essential because it only determines the fertility of the male sex. At the same time, it does not determine the male sex. The gene for femaleness is located on the X chromosome, and those for maleness are located on the autosomes. The gene for femaleness is located on the X chromosome, and those for maleness are located on the autosomes. When geneticist C.B. Bridges, working with Drosophila, crossed a triploid, that is 3n. Okay, diploid is referred as 2n and triploid is referred as 3n. So, when he crossed a triploid female with a normal male, he observed many combinations of autosomes and sex chromosomes in the offspring. Okay, we'll discuss what it is. From his results, Bridges in 1921 suggested that sex in Drosophila is determined by the balance between the genes for femaleness located on the X chromosomes and those for maleness located on the autosomes. We have learned earlier that genes responsible for femaleness is located on the X chromosome, and for maleness it is located on the autosomes. So what he says is, Bridges, that is what he suggested was, the Drosophila, the sex determination is determined by the balance between the genes for femaleness which are present on the X chromosome and the genes which are present on the autosomes, okay, which are responsible for maleness. So it is the balance between these two genes, that is genes on the X chromosomes and genes on the autosomes. Genes on the X chromosomes are responsible for femaleness, genes present on the autosomes are responsible for maleness. So this is the ratio between, or the balance between these two genes that determines the sex in Drosophila. This is what was suggested by Bridges. Hence, the sex of an individual is determined by the ratio of its X chromosome to that of its autosomal sex. Sex determination, it is determined by X chromosome, that is the ratio of X chromosome to that of its autosome sex. So this ratio is called sex index, and it is expressed as sex index is equal to number of X chromosomes divided by number of sets of autosomes, that is X divided by A. Okay, change in this ratio leads to a changed sex phenotype. Okay, we'll discuss what it is now. Bridges classical cross of a triploid female fly and a diploid male fly. Okay, he crossed a triploid female fly, that is with 3 autosomes plus 3 X chromosomes, and a diploid male with 2 autosomes and XY chromosomes. Okay, so this is the triploid female and this is the diploid male, and this is the parent's genotype, and these are the gametes, that is 3A plus XXX. So that results in gametes in the form combination of, that is 2A. Okay, so 1A is remaining. Okay, so 2A plus 2X chromosomes. So 1A plus the remaining X is the other gamete. So this is the way how gametes are formed. Okay, out of the 3 autosomes, 2 autosomes plus 1 autosome here. Out of the 3 X chromosomes, 2 X chromosomes combined with the 2A and the remaining X chromosome with this A. Or otherwise, it is 2 autosomes plus 1 X. Okay, reverse of this. 2 autosomes plus 1 X plus 1A with the other 2X. Okay, so in the same way, in diploid male, gametes are formed in like this. Okay, that is out of 2A, A here, and XY is 1X with 1A, the Y with another A. Okay, this is how this genic balance in Drosophila is determined. Okay, this is how it is formed, and this is a chart. It shows how this triploid female and diploid male forms individuals. Okay, please go through, you will understand. Okay, thank you.